Welcome to Chapter 16, the citric acid cycle. In this chapter, we will first consider the conversion of pyruvate, that is obtained from glycolysis, to acetyl groups, and then the entry of these acetyl groups into what we call as the citric acid cycle. The citric acid cycle is also called as the tricarboxylic acid cycle or the Krebs cycle. Next, we will examine the cycle of reactions and the enzymes that catalyze them. For most eukaryotic cells and many bacteria, which live under aerobic conditions, glycolysis is only the first stage in complete oxidation of glucose. Rather than being reduced to lactate or ethanol or some other fermentation product, the pyruvate produced by glycolysis is further oxidized. And it is oxidized to carbon dioxide and water. This aerobic phase of catabolism is called as respiration. And in this process, it consumes oxygen and produces carbon dioxide. This process provides more energy from glucose than glycolysis itself. It also captures energy stored in lipids and amino acids. Evolutionary origin, if you consider it, was developed about 2.5 billion years ago. It is used by animals, plants, and many microorganisms and occurs in three major stages. The first stage, production of acetyl-CoA from pyruvate. The second stage is the oxidation of acetyl-CoA. And the final stage, which we will talk about in Chapter 19, is the electron transfer and oxidative phosphorylation. It is in stage 2 that these electron carriers like FADH2 and NADH are generated, which enters the respiratory chain inside the mitochondria. Before understanding in detail, let us first briefly summarize the three stages here. The first stage of respiration involves acetyl-CoA production. And in this stage, it generates some ATP, NADH, and FADH2. And this is the stage where pyruvate gets converted to acetyl-CoA by the action of the enzyme pyruvate dehydrogenase. Carbohydrates release 1/3 of total potential carbon dioxide during this stage. In stage 2 of respiration, acetyl-CoA gets oxidized. And this is what we typically call as the citric acid cycle, because the reactions follow a cycle. This cycle generates more NADH and FADH2. 1 GTP is also produced. Remaining carbon atoms from carbohydrates, amino acids, and fatty acids are released during stage 2. Stage 3 of respiration is called as oxidative phosphorylation. And in this stage, the electron carriers that are generated in stage 2 enters the mitochondria and leads to the production of ATP. And this is the stage where a vast majority of ATP is generated. The citric acid cycle is also called as a TCA cycle or tricarboxylic acid cycle. It is also called as the Krebs cycle. So 3 different names. Respiration, the cellular process requiring oxygen and producing carbon dioxide. So in simpler terms, when you inhale oxygen, that oxygen is being used to convert glucose to carbon dioxide. And that carbon dioxide is what you are exhaling out. The tricarboxylic acid cycle is the second stage of cellular respiration and major point of carbon dioxide production. The real question is, why bother? Why do cells need TCA cycle or oxidative phosphorylation? The answer is because only a small amount of energy available in glucose is captured in glycolysis. Glucose is only partially oxidized. So if you see glycolysis, only minus 146 kilojoules per moles of energy is released, and carbons are still intact, 2 pyruvate molecules, and these carbons have lots of energy. The full oxidation of glucose would release about 2840 kilojoules per moles of energy, or 2840 kilojoules per moles of energy in complete oxidation, which results in formation of 6 carbon dioxide and 6 water molecules, while utilizing 6 oxygen molecules. In eukaryotes, the TCA cycle occurs in the mitochondria because the enzymes that are involved in this tricarboxylic acid cycle are expressed inside the mitochondria. Glycolysis occurs in the cytoplasm, that is outside the mitochondria within the cell. Citric acid cycle occurs in the mitochondrial matrix, as shown here. Oxidative phosphorylation occurs on the inner membranes. As you can see, that's where the oxidative phosphorylation happens. Except succinate dehydrogenase, which is located in the inner membrane, the TCA cycle happens in the matrix. And once the NADH is produced, those go to the inner membrane to participate in oxidative phosphorylation. Let us first consider stage 1 of respiration, that is acetyl-CoA production. Let us look at the enzymes that are involved and the reaction that is catalyzed. In the first stage, pyruvate is converted to acetyl-CoA, and this is accomplished by the enzyme pyruvate dehydrogenase complex. As you can see, the delta G0 for this reaction is minus 33.4 kilojoules per moles, and a molecule of carbon dioxide is released. The net reaction is the oxidative decarboxylation of pyruvate. First carbons of glucose to be fully oxidized and released as carbon dioxide. This is catalyzed by the pyruvate dehydrogenase complex, or PDC. This requires 5 coenzymes: TPP, or thiamine pyrophosphate, lipolysine, and FAD are prosthetic groups. NAD+ and Coenzyme A are co-substrates or cofactors in this case. The structure of coenzyme A is shown here. We briefly saw the structure of this cofactor in chapter 13, if you remember. And if you remember the structure, the business end of this molecule is this reactive thiol, which is in the form of a mercaptoethylamine attached to pantothenic acid, which is further attached to a phosphoadenosine diphosphate molecule. Now, the way this reactive thiol group works is by carrying an acetyl group. So hence acetyl-CoA, and the CoA is basically all these structures. Coenzymes are not a permanent part of the enzyme structure, and I'm sure you know this. They associate, fulfill a function, and dissociate. The function of CoA is to accept and carry acetyl groups. and that's what it does. Pyruvate dehydrogenase complex is a large multi-enzyme complex, up to about 10 megadalton. That is huge. This means 10 by 10 to the 6 Dalton in size. And this complex, isolated from mammals, is about 50 nanometer in diameter, more than 5 times the size of an entire ribosome and big enough to be visualized with the electron microscope. Now there are 3 enzymes that are involved in this complex: pyruvate dehydrogenase, dihydrolipoyl transacetylase, and dihydrolipoyl dehydrogenase, given by E1, E2, and E3. And if you look at the structure that is shown here, you can see these individual enzymes, E1, E2, and E3. Now this is a 3-dimensional image of the PDC showing the subunit structures. Now this image is a reconstructed image and analyzed from the cryo-electron microscope image. This is not a crystal structure. This is from a different technique called as cryo-electron microscopy. And one of the reasons why cryo-electron microscopy is used to determine the structure is because of the size of this complex. Anything about 200 Daltons, it's incredibly difficult to crystallize. And that is why cryo-electron microscopy is used to determine the structure. These are some of the advantages of multi-enzyme complexes. Short distances between catalytic sites allows channeling of substrates from one catalytic site to another. Substrate channeling, as they call it, minimizes side reactions. Regulation of activity of one subunit affects the entire complex. Let us consider the sequence of events in oxidative decarboxylation of pyruvate to acetyl-CoA. Step 1 and step 2 of the sequence is catalyzed by enzyme 1, pyruvate dehydrogenase. In step 1, decarboxylation of pyruvate to aldehyde happens. And here, pyruvate reacts with the bound thymine pyrophosphate of pyruvate dehydrogenase and is decarboxylated to hydroxyethyl thymine pyrophosphate. In step 2, the transfer of 2 electrons and the acetyl group from TPP to the oxidized form of the lipolysyl group of the core enzyme dihydrolipoyl transacetylase happens. And this results in the formation of acetyl thioester of the reduced lipoyl group, as shown here. Step 3 of the sequence is catalyzed by enzyme 2, dihydrolipoyl transacetylase. And in this step, the enzyme catalyzes a transesterification reaction in which the thiol or the SH group of CoA replaces the SH group of E2. Essentially what's happening is the acetyl group is transferred from this thiol to the coenzyme A, and the coenzyme then takes the acetyl group and becomes acetyl-CoA. And in the process, it is fully reduced and lipoyl group then goes back and becomes reduced lipoyl lysine. Step 4 and 5 of the sequence is catalyzed by the enzyme dihydrolipoyl dehydrogenase. And in step 4, reoxidation of the lipoamide cofactor happens. This enzyme promotes transfer of 2 hydrogen atoms from the reduced lipoyl group of E2 to the FAD cofactor. and FAD gets converted to FADH2, which then converts NAD plus to NADH and a proton. In step 5, regeneration of the oxidized FAD happens, and that is done in presence of the NAD plus that is waiting to accept electrons from it. So the net result of acetyl-CoA production is this. 2 pyruvate molecules reacts along with 2 NAD plus, resulting in 2 acetyl-CoA, 2 carbon dioxide, and 2 NADH molecules. And 2 pyruvate, as you know, is obtained from 1 glucose molecule. 2 acetyl-CoA that is produced in the reaction move on to the tricarboxylic acid cycle. The 2 NADH produced here move on to electron transport chain. This is the first step where carbon dioxide is released as a metabolic byproduct. Oxidation of acetyl-CoA happens during the tricarboxylic acid cycle. Leininger showed that the entire set of reactions in the TCA cycle occur in the mitochondria. So this is a snapshot of the citric acid cycle. And as you can see on the top, acetyl-CoA enters the cycle and it goes through the whole process. And the reason why it's called a cycle is because the final step of the cycle, where oxaloacetate is produced, is further used up along with acetyl-CoA to make metabolites again. And the result of the cycle is the energy extraction steps. And as you can see, there are 1, 2, and 3 NADH are produced along with 1 FADH2 and 1 GTP in this process. So the net result of the citric acid cycle, as I said, is 1 acetyl-CoA molecule along with 3 NAD+, 1 FAD, and 1 GDP produces 2 carbon dioxide, 3 NADH, 1 FADH2, 1 GTP, 1 CoA, and 3 hydrogen molecules. Net oxidation of 2 carbons to carbon dioxide happens in citric acid cycle. This is equivalent to 2 carbons of acetyl-CoA, but not the exact same carbons. Energy captured by electron transfer to NADH and FADH2 happens. Again, generates 1 GTP, which can be converted to ATP. Like I said, GTP is an ATP equivalent.